right, good evening, everybody. We have some learning targets to go over. I almost forgot to make this video, and I know that a lot of you count on me kind of talking through our learning objectives. So here goes. It's 8:50 at night. I'm supposed to be in bed. No, I never get to go to bed this early, but let's get started. Okay, learning objectives, things that we need to talk about. First of all, the first part of this unit is about meiosis. So it says, explain how meiosis results in the transmission of chromosomes from one generation to the next, that it ensures the formation of haploid, half the DNA, gametes, those are sex cells, in sexually reproducing diploid organisms. So in my body, every single cell in my body is diploid. That means it has pairs of chromosomes. I've got 23 chromosomes from mama and 23 chromosomes from papa in 100 trillion of my cells, except for the cells in my ovaries. Those are the egg cells, and those have a total of 23 chromosomes in each one. Those are haploid. All the other cells in my body are diploid. Let's just see what it says here. Meiosis results in daughter cells with half the number of chromosomes. Good. Meiosis involves 2 rounds of a sequential series of steps. We call them meiosis 1 and meiosis 2. Let's just talk through them really quickly. We've got it's IPMAT, PMAT when we go all the way through it. And let's see if I can show you this right here. We'll jump to it. So interphase, always interphase. That first cell is called a germ cell. And I know that's a weird word, but I always remember the G. So germ cells are what eventually lead to the formation of gametes. So there's a germ cell here where it says interphase. Then it goes into prophase. During prophase, super important things happen during prophase. And I think if I click on this, it'll bring, doesn't it bring me to the next part? Yes. Okay. So during prophase is when crossing over happens. So we see the pairs of chromosomes, they line up next to each other, the homologous pairs. They're called homo because that means same. So homologous pairs, you've got one from mom and one from dad. They carry similar genetic information. Maybe both of these have a trait for eye color, but maybe my mom's says my eyes are going to be blue and my dad says my eyes are going to be brown. They're similar chromosomes, not identical, but similar. When they line up or find their partners find their partners during prophase, they do something called crossing over. This is where they're going to actually trade pieces of DNA. And this is a place where we can get brand new genetic information. This adds to the diversity that's found within our gametes. Our gametes are super, super unique, and this adds to it. So what you see right here are 2 pairs of chromosomes joining at a chiasma and then exchanging little sections of DNA so they have new pieces of DNA. And then if you look down at the bottom here, you see we've got the original chromosome where no crossing over took place, but then we've got 2 new chromosomes where crossing over did take place, and then an original chromosome where nothing crossed over. So we have actually 4 unique gametes in this example. Those are called recombinant chromosomes. They have recombined during crossing over, and that becomes really important later on when we start looking at probability and why sometimes our statistics are off a little bit. We predict that something's going to have a certain ratio, and then the ratio doesn't work exactly the way we expect. And that's because there's been some crossing over and linked genes have been separated, or separated genes have come together. All right, so here's prophase again, chiasma, crossing over. When they find their partners, that's called synapsis, and it doesn't actually have that word here, but just finding their partners is called synapsis. Then they make a little chiasma, and then they cross over. So then the next stage is metaphase, and metaphase 1 of meiosis is super unique because they line up with their partners. So you've got 23 chromosomes on the left and 23 chromosomes on the right if this were taking place in a human. Then during anaphase, the partners separate, but they are all still bivalent. So look in the diagram and notice that these are little, what kind of look like X's being pulled to the sides. They are bivalent. Then the cell goes through cytokinesis, and we actually cut the chromosome number in half. 23 chromosomes went to the top in this diagram, and 23 chromosomes went to the bottom in this diagram, and then the cell divided, and so we have 23 chromosomes. Why aren't we done with cell division? Because all of those chromosomes are bivalent. That's more than necessary in terms of how much DNA. So we're going to go through the next division again to make all of those chromosomes monovalent. So then we go through, give me just a second. So we did metaphase 1 already. I'm trying to think if there's anything you need to know about that. I don't think so. And now there's no DNA replication or not another interphase. So it's just IPMAT, PMAT. So now we split it. We have 2 cell lines because we just split them in 2. So this time there's no crossing over that happens. During metaphase 2, they line up single file instead of with their partners. Why don't they line up with their partners? Because their partners are in the other cell now. So the partners are away from them. Then anaphase, the bivalent chromosomes get ripped in half. So now it's a monovalent chromosome going to each side. Remember that the monovalent chromosomes are supposed to be identical copies of each other. However, because of crossing over, they're not always identical to each other, and that's adding to our genetic diversity. And finally, in that last stage of telophase 2, the cell divides again. So what we have at the end are 4 haploid gametes. If this happened in a female, these would be 4 eggs. And I will tell you that only one of the eggs survives, and it collects almost all of the cytoplasm, and then the other eggs disintegrate. You didn't need to know that, but I'm just telling you that. If it happened in a male, then these would be 4 sperm, and all 4 sperm survive. If this happened in a flower, it could be eggs. If this happened in a flower, this could be pollen, just to give you a sense. All right, so let's go back to our learning targets and see what else we need to know. Describe similarities or differences between phases and outcomes of mitosis and meiosis. So to be really clear, so we've talked about the differences in the phases. Let me go back actually and just glance at this quickly. So this right here, this meiosis 2, all these M words, meiosis 2 is very similar to mitosis. Notice that in mitosis, they line up in the middle, the bivalent chromosomes get ripped apart, and they split in 2. They're very similar. Only if this is happening in a human, there's half the DNA here. If this was mitosis, there would have been 46 chromosomes lining up, but because this is meiosis 2, there's only 23 chromosomes that are lining up in the middle. So that's where we're talking about the similarities and differences. Now, what's super important is you understand what we end up with. At the end of mitosis, we've cloned our original cell. So our original cell had 46 monovalent chromosomes. During the S phase, those monovalent chromosomes were copied, and now we have bivalent chromosomes. Then during mitosis, they get separated, and the cell divides, and we end up with, again, 46 monovalent chromosomes. We've just cloned the original cell. But in meiosis, and those cells for mitosis, they were diploid because they had 23 from mom and 23 from dad. But the 4 cells that we get at the end of meiosis, they're haploid. They have half the DNA. Of course, they're monovalent. We separated them from their partners. And each one is slightly genetically unique. And that becomes really important in just a minute. All right. Okay, good enough. Explain how the process of meiosis generates genetic diversity. And you need to know, I think these are the 3 right here. And they say them right here. So you need to know these. Like if you should be able to say how genetic diversity is increased during sexual reproduction, for example. So the first thing it says, separation of the homologous chromosomes in meiosis 1, so them being pulled apart from their partners, ensures that each gamete receives a haploid set of chromosomes that comprises both maternal and paternal chromosomes. So what that means is Mendel's law of segregation says that they can line up on either side and be separated from each other. So you're not always getting mom's chromosomes on one side and dad's chromosomes on the other. So that means that the sperm and egg that are forming are a combination of mom's chromosomes and a combination of dad's chromosomes. Then another source of genetic variation, the crossing over. So they form the chiasma, they cross over, they trade pieces of DNA. So we're creating chromosomes that have never existed in the world. Not only have we have a combination of mom's and dad's chromosomes, but those chromosomes that are there, some of them are totally unique because they're little pieces of dad's and little pieces of mom's, and they never existed before. Then the last way that we get genetic diversity has to do with random assortment. So sexual reproduction in eukaryotes involving gamete formation, including crossing over, the random assortment of chromosomes during meiosis, and subsequent fertilization of gametes serves to increase variation. So the subsequent fertilization of gametes, remember some of those statistics, and I think that's in this set of notes here. Let me see if I can fly through it really quickly. These numbers right here. So remember that how many possibilities? So there are more than 8 million possible combinations of our 46 chromosomes in any given gamete. So crazy number of variety of gametes. Plus we have crossing over. Plus we multiply the odds of the sperm times the odds of the eggs, which sperm and which egg are going to come together. So any zygote, that might be a word you have forgotten. Zygote is sperm and egg coming together. The odds of any one sperm and one egg, you multiply them together. So there's more than 70, is that trillion? 1,000 million billion trillion, 70 trillion possible combinations. And so that's more than all the human beings that have ever lived on the face of this planet. In just one or couple, in just two people, they can produce that many diverse human beings. And that didn't even take into account crossing over, which added even more genetic variation. Okay, hopefully that made sense. Let me know if you have questions about that. All right, oops, that is not what I meant to do at all. Number 4, the inheritance of genes and traits as described by Mendel's law. Mendel's laws of segregation and independent assortment can be applied to genes that are on different chromosomes. So let me show you that, because I think we can show you those two right here. Give me just a second. Sorry, it takes a moment to get there. I thought segregation means to separate, and I thought they were right here. Oh my gosh, I don't mean to slow us down. Sorry, people. I am not seeing where it talks about segregation and law of independent assortment. Well, you're just going to have to trust me, folks, if I can't find them, that the law of segregation says that the chromosomes are separated from each other, and independent assortment says that they don't line up on the same side of the metaphase plate. They don't line up. So sometimes mom is here and dad's is here, and sometimes they're on opposite sides. And then they get separated or segregated from one another. I thought it was right in that set of notes. I'm missing it. My apologies. All right. B, fertilization involves the fusion of two haploid gametes, forming a zygote, which is a fertilized egg. And rules of probability can be applied. Okay, moving on. Okay, pattern of inheritance. You need to know monohybrid, dihybrid, sex-linked, genetically linked. So monohybrid is just the 2x2 box. Dihybrid is the 4x4 box. However, we learned, and you can do much more complicated things than dihybrid if you multiply the probabilities. So remember that for a dihybrid, you could do a 4x4 box and have 16 letters, 16 boxes of letters to fill in, or you can do 2 monohybrid boxes and multiply the probabilities. And I strongly recommend that, because on the test, you will almost certainly be given something way harder than a dihybrid. I'll probably give you a pentahybrid. Is there such a word? I can't remember. Like 5 different traits, and you just need to do the monohybrids on all of them and multiply them together, and that will give you your probabilities. Okay, can often be predicted from data, including the pedigree. So you need to be able to read pedigrees and predict whether they are, or identify whether they are dominant recessive or sex-linked recessive. They give the parent genotype and phenotype. So remember, genotype is the letters and phenotype is what it physically looks like. So somebody's genotype who has sickle cell trait might be AS, that's their genotype, and then their phenotype is that they have sickle trait. Give me just a second, I have to pause for just a moment. Sorry about that. My dog was getting into trouble. My family is due home any minute. They're going to interrupt me too, so I'll have to pause this a few times. All right, let's see. So, and then phenotype, again, is what they physically look like. Let's move on here. I don't know why that's still there. There we go. All right. Explain deviations from Mendel's model of the inheritance of traits. Patterns of inheritance of many traits. So let's do incomplete versus complete dominance. Incomplete leads to blending, and you would use 2 capital letters for that. So that's where we talked about like a red flower mating with a white flower and forming a pink flower. So the genotypes would be big R, crossed with big W, produces big R, big W. That's the pink. Versus codominance, which is like the A and B alleles for blood type. They are codominant. So when you get one from mom and one from dad, they both show up. So if you get a type A from mom and a type B from dad, you end up with type AB blood. And that's, you have both the A and the B traits on the outside of your blood cells. All right. Genes that are adjacent and close to one another on the same chromosome may appear to be genetically linked. The probability that genetically linked genes will segregate as a unit can be used to calculate the map distance between them. You will be asked to calculate the map distance. Let's see if I can remember. I have a problem where you can look at it. Give me just a second. Oh, it's going slow. No time for it to go slow. There we go. So this is the exact kind of problem that you're going to be asked to solve on the test. You'll be given a series of a series of traits and how often they cross over and get separated from one another. And so that tells you roughly how far apart they are. So if you see a number that's really, really, really big, that means that they're crossing over, they're separating from each other a lot of the time. So that means you would put them farther apart on your chromosome map. But if you see that it's like a 1% or a 2%, then you're going to put them really close together on your chromosome map. So you got to be able to do that, and let me know if you have questions about that. All right. So map distance is the same as the percentage of times that they cross over. So if it says 28%, then they are 28 map units apart. Some traits are determined by genes on sex chromosomes and are known as the sex-linked traits. For the most part, every time that you see a sex-linked problem, it is going to be a trait that is carried on the X chromosome, but technically anything carried on the Y chromosome is also a sex-linked trait. And almost always it's going to be a sex-linked recessive trait, but not always. If it's sex-linked recessive, it's more common in males, and that's because females have that backup copy. And so males get one copy, and if that copy happens to have the genetic trait that we're studying, then they automatically have it. So we gave the example of pretending like red-green color blindness. If we say it's 1 in 25 chances of getting that trait on the X chromosome, then a guy has a 1 in 25 chance of developing red-green color blindness. However, for a female, in order for her to have red-green color blindness, she has to have 2 copies of it. So that's 1 in 25 times 1 in 25. So 1 in, what's 25 times 25? Is that like 625 or something? I don't know, whatever. So her odds of developing red-green color blindness are so much less than they are for a male. Okay, hopefully that was good enough. If one of the chromosomes of the female randomly inactivates, this is called a bar body. So when X chromosomes disappear or stop working, that's called a bar body. And you can look at that if you're interested. Okay, moving on. Many of the traits are a product of multiple genes. They're called polygenic. There are so many things in genetics that are not just as simple as we make it sound when we're first introducing Mendel's genetics. So for example, skin color, there's this huge rainbow of brown colors. It's not just white or black. And so that is because we have many, many, many genes. Like, there are lots of estimates. I think the most recent I've heard is 6 to 8. No, that was cancer. I don't remember, but lots and lots of genes that are controlling our skin color and how much melanin is being produced. Same thing with height, polygenic. There's not just short and tall people. We got all these heights in between, many genes controlling our height. Eye color, polygenic, lots of things. So those are the three I think you should know. Skin, height, and eye color. If those are on the tip of your tongue, you're doing pretty well. All right. Some traits result from non-nuclear inheritance. You need to know this. Chloroplasts and mitochondria, we think that they were once bacteria, remember? And they have their own DNA. So they have traits, which, we don't talk about as much. They have their own characteristics that get passed on, and it has nothing to do with our nucleus. So we call that non-nuclear inheritance. Now, what's important is I mentioned just a few minutes ago that when a woman's body, when a female, I shouldn't even say woman, it can be any female organism, goes through meiosis and produces eggs, only one of the eggs survives and the other eggs disintegrate. That one egg contains all of the organelles. The sperm are just packets of DNA. That's all they are. And so it is the egg that's transferring the mitochondria and the chloroplasts. So when we look at mitochondrial DNA or chloroplast DNA, we're talking about the female's inheritance, and we're looking at the female side of the family, not the male side of the family, because the male sperm is just carrying the DNA itself. Okay, in animals, mitochondria are transmitted by the egg and not by sperm. In plants, mitochondria and chloroplasts are transmitted in the ovule, which is an egg in a plant, and not in the pollen. Good, maternally inherited. Okay, explain how the same genotype can result in multiple phenotypes under different environmental conditions. So this is called phenotypic plasticity, and unfortunately, I gotta think where that was. I think that's this. Nope, one more. The word phenotypic plasticity was not used in our discussion here. So in this Prezi, phenotypic plasticity is like the idea that these are genetically the same flowers, but they look different depending on the soil that they're growing in, the pH of the soil that they're growing in. This could be the same rabbit. It's not, but it could be the same Arctic hare, but their coat color is dependent on the environment. It's dependent on the season of the year. And there's many, many other examples. Height we said already that height is polygenic, but height is also plastic. Height also is determined by your environment and your nutrition. And so poor nutrition leads to lower height. All right, so that's phenotypic plasticity. Next, explain how chromosomal inheritance generates genetic variation. We already talked about that. Chromosomal basis. Okay, certain human genetic disorders can be attributed to the inheritance of a single affected mutated allele or specific chromosomal changes, such as non-disjunction. Gosh, guys, I don't remember where non-disjunction is mentioned. Is that in this one? There it is. We definitely need to go over non-disjunction because this was a problem in students last year when they went to take the test. They bombed this. So please pay attention to this diagram. Let's make it up close. So what's supposed to happen in meiosis? There's two ways that we can have what's called non-disjunction. I hate this word. You just have to know it. It's not a word that intuitively they're, oh yeah, I know what that means. You don't. So remember that in meiosis, so this would be like anaphase of meiosis 1, the chromosomes were sitting next to each other, and now they're supposed to go to opposite sides. So if it were happening in a human being, 23 would go to the left and 23 would go to the right. In this cell, there's just 4 chromosomes. So 2 bivalent are supposed to go to the left and 2 bivalent are supposed to go to the right. But instead, 2 of them stay stuck together. So we actually end up with 3 bivalent chromosomes going to the left and only 1 bivalent chromosome going to the right. Then as we go through into meiosis 2, during the next anaphase, they pull apart. So this one Now I'm going to scooch it down. Now there's only one bivalent chromosome, and it gets ripped apart into 2 bivalent chromosomes. And these 3 that were there, they get ripped into 3 monovalent chromosomes. And what you end up with at the very end, these are the sperm or the egg or the pollen, is really weird ones. You end up with these gametes have an extra chromosome in them. They're supposed to have 2 because the original cell had 4. So they have 3 in them. They have an extra one, and these are all missing one. So that's one form of non-disjunction. The other form of non-disjunction, instead of taking place during meiosis 1, can take place during one of the cells' meiosis 2. So notice that this developed correctly. Meiosis 1, they went their opposite ways. It's all perfect. On the right side, this one split, the bivalent chromosome split perfectly. However, in this cell here, instead of one going to the left and one going to the right and one going to the left and one going to the right, these guys stayed stuck together as a bivalent chromosome. So now 3 chromosomes move to the left and only 1 moved to the right. And what you end up with is one of our sex cells, one of the gametes ends up with an extra chromosome, one ends up with a missing chromosome, but then 2 have the correct number of chromosomes in them. So in this example, again, the germ cell, practicing that word, the germ cell had 4 chromosomes. The gametes are supposed to have 2 chromosomes. So this is all non-disjunction. And for the most part, in vertebrates, and many different types of animals, you have to have, the organism has to have the right number of chromosomes. Having the wrong number of chromosomes is often fatal, almost always leads to a syndrome that has some kind of a genetic disorder and usually major disabilities. So having the wrong chromosome number is super, super bad for the organism. And I just want you to think this through really quickly. Let's say we had a sperm that had an extra chromosome in it, and it joined with an egg that had the correct number of chromosomes. You would end up with an individual that had 47 chromosomes. If that were chromosome 21, that we had the extra one, then that's Down syndrome. Down syndrome is called trisomy 21. 3 of chromosome 21. The reason trisomy 21 is so survivable is because chromosome 21 is very, very little. So having a third one doesn't seem to impact the organism as much as some of the larger chromosomes. If we have 3 of those, that's almost always leads to the miscarriage of the child. All right, let's go back. Non-disjunction. Okay, so errors in mitosis or meiosis can result in change in phenotype. So triploidy would be where we get, where an organism has, I was going to try to see if there's an image on this. Triploidy is where an organism has 3 of all the chromosomes. And let me see if, there we go. Here's what we were looking for. No. Come back. That's what we want. Okay, so this is a haploid organism. They have 1 copy of every chromosome. So this is like our eggs or sperm. Diploid is all the other cells in our body. We have 2 of all of our chromosomes. You get half of them from mom and half of them from dad. But there are some organisms that are polyploid. That means they have 3 or more sets of chromosomes. We can't survive that way. But some organisms like plants, I'm drawing a blank now on all the different organisms. I think I have it written here of all the different things. So commercial fruit, fish, and mollusks. There we go. Those are examples. Are organisms that can benefit from having extra chromosomes. So this is a confusing learning target. Changes in chromosome number often result in new phenotypes, okay, including sterility caused by triploidy, I got to explain that, and increased vigor of other polyploids. So I don't think we covered that a whole lot in the other things. So I added these little paragraphs. So let's just go through them here really quickly. Vertebrates generally do not tolerate polyploidy. That means that we have to have diploid cells, and if you give us even 1 extra chromosome, we have a problem. However, the high incidence of polyploidy in plants, frogs, and fish indicates that there are advantages to this condition. Triploids are widely used in both commercial fruit, fish and mollusk production, as they can have beneficial commercial traits such as, so here's your increased vigor, improved growth, pathogen resistance, and unfortunately, infertility, conferring protection of elite genetics. Oh my gosh, what did that all mean? This means that commercial farmers of these organisms control reproduction to prevent organisms that have developed unwanted mutations from mating and passing on their DNA. So farmers are super, super particular, and I'm not just talking about farmers of corn. We're talking about all of these kinds of organisms. Once you get the ideal organism, you want to clone it, basically. You want it to reproduce and reproduce and reproduce and reproduce. But it turns out that in this case, they're sterile and they have to be hand reproduced by the farmers. They have to control reproduction, but they have all these great traits in them as well. So somehow triploidy or polyploidy is beneficial to organisms, but it can also lead to sterility, which is a weird thing. Last paragraph here. Polyploidy appears to prevent the accumulation of recessive mutations in the genomes, the DNA, of later generations, thereby maintaining hybrid vigor. So they are stronger and healthier because they have so many extra chromosomes. Kind of like those backup X chromosomes that females have. It gives us a little bit of protection. This is more so because they have backup copies of all of their chromosomes. Another important factor is gene redundancy. Because the polyploid offspring now have twice as many copies of any particular gene, the offspring are shielded from the deleterious effects of recessive mutations, similar to the benefit that females have over males by having a backup X. Okay, and you can read more about those if you're interested in them. I was surprised that those were listed as learning targets because we don't usually cover that information. Changes in chromosome number often result in human disorders with developmental limitations, including Down syndrome, trisomy 21, and Turner syndrome. Let's see, Turner syndrome, I think, is that in this one? Turner syndrome is females that are XO, and I can't remember, is that this? Nope, that is not this set of notes. Females that are XO, that means that they are missing an X chromosome, so they have only 45 chromosomes. Here we go. And then another one that's super common, so there's Turner syndrome. It's a survivable condition because of Barr bodies. A lot of times the second X chromosome shuts down, so you can survive with just one X chromosome. The other one that's a very common this is a trisomy, is Klinefelter syndrome. And this is boys that they have the XY, they have all the boy parts, but they have an extra X chromosome. And so they tend to have more estrogen, which leads to some female characteristics like higher voices, lack of beard growth, and then breast development, unfortunately, which is often very traumatizing for young men. All right, we'll go with that. Back to learning targets. Determine whether or not the results of genetic crosses between two genes support the null hypothesis that the genes are not linked. So here's the chi-squared equation that you need to know. And it says unlinked traits should show a 1-to-1-to-1-to-1 ratio, but linked traits should show a 1-to-1-to-0-to-0 ratio. So there's no recombinance, there's no combinations that are different than the original parents. Let's review. Give me a second. I want to find it. Let's review that in Mendel's genetics. And was it beyond this one? Give me a second. Here we go. This is what we're looking for. So these are the problems that nope, almost. These are the problems that we were looking for right here. So if the genes are unlinked, if they're located on different chromosomes, then they should show a 1-to-1-to-1-to-1 ratio. What are we talking about? We're saying that you would have the ratio of the parents would be equivalent to the ratio of ones that are crosses of the parents. So notice that this parent is gray body and normal wings. So here are offspring that are gray body with normal wings. Then the other parent is black body with vestigial wings, little wings. Here's offspring that look just like that parent. But what they also would have if the trait for body color and wings are on different chromosomes, you would also get combinations where we have a gray body but little wings, or a black body with long wings. And according to probability, we should get those in a 1-to-1 to 1-to-1 ratio. And that's how we know that they're not linked. If they're on the same chromosome and that chromosome travels with both of them attached, then we should only see one offspring that either look like the mom or offspring that look like the dad, and no recombinants. However, we find that sometimes we get ratios where we get something that's sort of in between. Why are we getting something that's sort of in between? It is because they are, in fact, on the same chromosome, but occasionally it crosses over and they get separated as though they are on different chromosomes. Only they're not. They were originally on the same chromosome, and now they end up on different chromosomes. And so instead of seeing this 1-to-1 to 1-to-1 ratio, or 1-to-1 with 2 zeros, we get something that's sort of in between. And that's an indicator of crossing over. And then we use that information to calculate map distances. So that's where we did the 206 plus 185. Those are the recombinants. So these two numbers added together, divided by the total number of offspring, was 17%. And so we learned that they were 17 map units apart on the chromosome, and that's why they separated 17% of the time. And then you can calculate a little bit more information than that once you add in another chromosome, or add in another trait, sorry. Okay, so hopefully that made sense. However, crossing over linked traits will create some individuals with non-parental phenotypes instead of, and I give you an example here of what that would be. Okay, moving on. Was that it? Well, apparently that was all of it. I didn't know we were almost done, you guys. So please let me know if you have questions. I didn't realize we were on the last one. So the one thing I will say, just then, as long as I have a moment here, this chi-squared problem, for sure review the making of the fittest, that chi-squared bunch of questions that you had at the end about sickle cell anemia. Review those, because there will definitely be a question similar to that on the test. Okay, let me know if you have any questions.